So today we're going to start talking about chapter 16, and we'll probably just do kind of the first part of this. One of the important things about this chapter is it's going to start introducing the ideas of the immune system, and it's a little bit tricky. One of the hard things about like the body is that everything kind of works together, and so sometimes it's hard to like separate one system and just say, oh, we're only going to talk about this. And so in this part particularly, it really merges and meshes a lot with the immune system. And so we'll talk a little bit about that as well. And there's a whole nother chapter on the immune system, but we might kind of introduce some of those ideas here. But as far as this chapter goes, the primary topic here is about the lymphatic system. And the lymphatic system has a couple of different functions and purposes, but two of the main functions and purposes of the lymphatic system are, one, it helps clean the blood and clean the fluids in our body to protect us from infections or keep them from spreading through our body. And then the other thing that the lymphatic system does is it also helps to it also helps to return the fluids in our periphery back to our central circulation. And so last time we kind of talked about the heart and how it beats, or maybe 2 times ago, I can't remember, but it pushes the blood away from your body. When it gets to your hands, your feet, and stuff like that, then fluids go there. And we have to have other ways to get the blood back. So we talked about muscle contractions, we talked about pressure changes in the thoracic cavity and things like that, the squish veins, and all those things help. But the lymphatic system is going to help with that as well. So the lymphatic system will all help to pick up fluids that are in our periphery and bring it back to the central circulation. So let's pop into this first slide here. So like we just talked about, the main functions of the lymphatic system, it's going to transport fluid away from like the periphery back to our bloodstream. It's going to absorb, we'll talk about that in a second. It's also going to fight off diseases and protect us from diseases. We'll talk about that. And that's the immune system part. And then the other thing that it does that it's really important for is it helps transport lipids. So in the digestive system, there are special lipid transporting things called lacteals, and they absorb fats from our digestive system and help move it through our bodies. And so that's one of the other main functions of the lymphatic system. So this picture here just kind of illustrates what we were just talking about. So the heart pumps and it pushes the blood to the periphery. When the blood gets to the capillaries, some of the fluids of the blood actually leak out of the capillaries and get into the interstitial space. So in between the blood vessels and the actual cells of our body, there's small spaces, and fluid can leak out and get in those spaces. And so the lymphatic system will pick that up. And so to kind of illustrate this point, there's a parasitic infection that causes what's called elephantitis. And people that get elephantitis, they get these parasites, and the parasites grow inside the lymphatic vessels, and they block the vessels. And so people that have that happen, then they get a whole bunch of edema in their like appendages, like their legs and stuff, and their legs just look like a giant tree trunk. And that just kind of illustrates the idea that the lymphatic system under normal circumstances will return that fluid, but if it's blocked, it won't work. Another example to kind of illustrate that part of the lymphatic system is around the pectoral region of our bodies. There's a lot of lymphatic tissue there, and in the axillary region of our bodies. And women who have breast cancer, if they have like a mastectomy or they have to have like a lump removed or something like that, a lot of times they also remove lymphatic tissue. And so in that case, a lot of times what happens to women is on the side of their body where they have that tissue removed, they get lymphedema in their arms. And so that's something that's very common in people that have had tissue removed, lymphatic tissue removed. And so that just kind of helps illustrate that idea. Okay, so the lymphatic system can help pick that up. So similar to the blood vessels, what we're going to see in the lymphatic system is we're going to have small capillaries that intermingle with the capillaries of the blood vessels, and they'll pick up fluid and they'll bring it back towards the central circulation. And as they come back towards the heart, the vessels are going to keep getting bigger and bigger and bigger, just like the blood vessels do as they come back to the heart. So we're going to have lymphatic capillaries, and then we're going to have larger vessels as they come back. So capillaries, lymphatic vessels, lymph nodes, we'll talk about those in a minute. And then lymphatic trunks, and then collecting ducts, and the subclavian veins. So these are really small, just like we said with the capillaries of our blood vessels. They're basically 1 cell thick. They're going to be just an epithelial cell, 1 cell thick. And then as they come together, they'll make larger vessels that are intermediate level, called lymphatic vessels. Then there'll be these lymph nodes that will help clean stuff along the way. We'll talk about those in a second. And then we'll come to larger lymphatic vessels, and then they come together to form trunks. And then the trunks come together to form collecting ducts, and then the collecting ducts dump into the subclavian veins. So there's 2 collecting ducts. And of course we'll talk about this more in a minute, but that's kind of the idea. And it just gets bigger, and it just merges together to come back to the blood. When we talk about the capillaries, we just mentioned they're 1 cell layer thick, so they're really small. Another kind of interesting feature of the capillaries is that they're not open-ended, so they're closed off on the ends. And the cells on the ends actually overlap. So they overlap, so as the fluid comes in to the lymphatic capillaries, it creates kind of like a valve. And so if pressure pushes it back, the fluid can't get out because it snaps those cells shut on top of each other, and it creates kind of a valve that prevents them from going back out, the fluid from going back out. And so the fluid can only flow in one direction. The larger lymphatic vessels also have valves as well, just like veins have valves. And so that also helps with the return of the fluid back to the central circulation. So we can pick up fluids here. The way the fluids are picked up, there's a couple different ways they're picked up. Primarily, the fluids get picked up through the lymphatic vessels because of the osmotic gradient. So some of the osmotic gradient is going to help in the bloodstream. Because you're losing fluids, it creates a concentration gradient. It will actually absorb some of the fluids back into the blood. But then the lymphatic vessels can also pick up fluid that way as well. And then because of this valve system that we just talked about, those are kind of the main ways that you pick the fluid up into the lymphatic vessels. This picture here kind of represents the lymphatic system in our bodies. So when you look at this, there's a really wide distribution of lymphatic tissue all over, and that's really important because, as we mentioned before, when the heart pushes the blood away from our central core, the further and further away the blood gets, the lower the pressure gets. And so when the pressure gets out here, when the blood gets out here, there's nothing to get the blood to come back or the fluids to get back unless we have these other resources like muscle contractions and pressure changes. And so the lymphatic system has to be distributed all over our body. What you'll notice also in this picture is that in certain places there are little spots that kind of look like little peas. And so that's what we're going to talk about next. Those are called lymph nodes. And lymph nodes are a very important part of the lymphatic system. And this is where we're going to start getting into the immune system and the protective or the cleaning part of what the lymphatic system does to help fight off infections or protect us from infections. Actually, we'll talk about this. We better talk about this first. So, as we mentioned before, there are 2 lymphatic trunks. So we'll talk about the immune system stuff in a second. I thought the lymph node was next. But there are 2 lymphatic trunks. The 2 lymphatic trunks are shown here. And what you notice is the dark red portion that's shown on this man's body, that is what the right lymphatic duct, I think I said trunks, I meant ducts. There are 2 lymphatic ducts. And the dark red portion that's outlined on this person emphasizes the distribution of the lymph and what's drained into the right lymphatic duct. And what you notice is it's not symmetrical and it's not the same. So the right lymphatic duct and the portion of lymph that it drains back into the blood is very different from the thoracic duct. So the 2 ducts, as these trunks and all these vessels come together, they eventually drain into the right lymphatic duct and the thoracic duct. The right lymphatic duct is going to drain lymph from the right arm, the right side of the chest, right armpit, and the right side of the face and neck. And the thoracic duct drains the lymph from all the rest of the body. So it seems kind of weird It's very different than other systems in our body. They're usually very symmetrical and they're evenly distributed and stuff, but this is one that's not. Yeah. Anyways, so those are the two main ducts. As the lymph is moving through the vessels, through the lymphatic vessels, because of pressure changes and skeletal muscle contractions, along the pathways of these lymphatic vessels, we're going to have these special immune system structures that are called lymph nodes. So lymph nodes are part of the lymphatic system, and the way that they're built is they're built so that the vessels going into the lymph node, or the afferent lymphatic vessels carrying the lymph into the lymph nodes, carry the lymph into these special compartments that are filled with white blood cells. And so the structure of them is that you're going to have an outer capsule that compartmentalizes the fluid and the stuff that's going in there. And that capsule then is split up, the space inside the capsule is split up with what are called trabeculae. So the trabeculae create kind of little rooms that are jam-packed full of white blood cells. They're full of T cells, B cells, and macrophages. And the orientation of these is that you see kind of a circular pattern here called the germinal center. And the germinal center has a couple of different things in it, but it's especially full of B cells. And then it's surrounded kind of like a donut that has some macrophages and some T cells in it as well. And so as the fluid comes in, because of these little compartments and the way that the space is broken up, the fluid is forced to go across these compartmentalized concentrations of white blood cells, of T cells, B cells, and macrophages. And so if there's any kind of pathogen or toxin or anything inside the lymph that is potentially dangerous for our body, before we go and dump it into our bloodstream, these lymph nodes just kill it, just get rid of it. They eat it, and they also will, because we'll talk about this more later, but B cells are what produce antibodies. And so then they also will create an immune response against any kind of pathogen that we might be collecting inside our bodies and help protect us from those in the future. So the lymph goes in there, the white blood cells kill all this stuff coming in, and then the fluid is forced to go through these little channels, and then it exits through the other side of the lymph node through the efferent or efferent lymphatic vessels. And so that's kind of the pattern that you see when you talk about the lymph node. They're really important, and what we'll find is that they're kind of distributed all over our body, but there are certain areas in our body that have really high concentrations of them. So in our neck, there's a lot of lymph nodes. There's a lot of lymph nodes in the armpits, in the axillary region, and then like the groin area, there's a lot of them in that area as well. And there's some other places also, but those are some of the main places where you find those lymph nodes. And so when we talk about the lymphatic system, there are different organs associated with the lymphatic system. And of course, one of them we just mentioned are the lymph nodes. Those help clean the lymph, help fight off infections and create immunity against potential diseases and stuff. Another kind of lymphatic tissue in our bodies is called the thymus. So the thymus is an organ. It's found in the mediastinum, just kind of, I guess I should do that, right on top of the heart. It's got two lobes, kind of looks like a little butterfly. And the important thing about the thymus, I mean, there's several things it does, but one of the main things it does, and for our conversation today, is the thymus is the location of T cell maturation. So T cells are a really important part of our immune system, and T cells and B cells both come from the bone marrow. They're born by hematopoietic stem cells in the bone marrow, we talked about a couple weeks ago. And in order for the T cells to actually work and fight off infections, they have to mature. They go through a selection process. And that selection process chooses T cells that will help fight off infections, but gets rid of T cells that potentially could cause self-harm. And so that happens inside the thymus. It's called antigenic, not antigenic variation, it's called, I forget what it's called. Anyways, it's a selection process that gets rid of T cells that can potentially cause autoimmune issues. So basically what happens is in our body, the DNA that clones for our T cells, our bodies just randomly put those T cells together in so many different ways. And so we create potentially T cells that can fight off like lots and lots of different kinds of infections. Our body doesn't know what kind of pathogens we're going to be exposed to in our lifetime. And some of the pathogens that we're exposed to at some point in our life might not even exist when we're born. And so there are lots of changes in the antigens that are around us as we progress through our life. And so our body creates these T cells against all these different pathogens that we might come in contact with through our life. The problem is that sometimes it also accidentally creates T cells that will target self-antigens. And of course we don't want that to happen because we don't want our immune system to attack our cells. And so it goes through the selection process where in the thymus it will expose the T cells to self-antigen, and if they become reactive to it, or if they get excited and start fighting it, then the body just kills them. So it kills the ones that are self-reactive and it keeps the ones that aren't. And then as we go through our lives and we're exposed to these different antigens, then our body can stimulate the T cells that they're sensitive to, and then part of that process will be that we'll make more of those kind of T cells to fight off those infections. And so the thymus is really important. When we look at this, you can see in this picture that the thymus is just sitting here right on top of the heart, a little bit above it, sort of in the top of the mediastinum. So that's important. Okay. Another kind of lymphatic tissue is the spleen. So the spleen basically has the same kind of structure as a lymph node. It's kind of like a big lymph node. And so it's bigger than a lymph node. The other main difference between the spleen and a lymph node is that the spleen actually processes blood instead of lymph. And so lymph nodes only are going to take lymph as it goes through the lymphatic vessels, but the spleen has splenic veins and splenic arteries that go to it and go from it that carry the blood to the spleen. So this is really great because if we do actually get an infection inside of our blood, or there's some kind of toxin or something that isn't supposed to be there, then one of the places where we can detect that and get rid of it is in the spleen. And the spleen will have broken up areas inside of it, similar to how we saw the lymph node. And these little sections are going to be just jam-packed with white blood cells. And so as the blood goes through the spleen and it's forced to go through these little areas, then the white blood cells will recognize any kind of pathogen or toxin. It also recognizes old red blood cells. So as red blood cells go through our body, they have a pretty short lifespan. And as they get old, they start to break apart. And so one of the places where we get rid of old red blood cells is in the spleen. So the spleen will recognize old red blood cells and break them down, eat them up, and then recycle the iron and the other important parts of the blood cell so that we can make more. And so the spleen is really important. One of the other things that's important to think about when you think about the spleen is because of its size and because it's collecting the blood, it holds a pretty high volume of blood at any given time. And so any kind of injury that can do damage to the spleen and cause the spleen to rupture can be life-threatening because suddenly you get this huge hematoma, this huge internal bleeding that can, of course, be life-threatening. People can live without the spleen, and there are interventions that you can go through if you do get into an accident where you're injured that way. But it's a really serious thing, and you have to be really careful if that happens. All right. Okay. There are other types of tissues that are associated with the lymphatic system, but those are some of the main ones, and those are going to be the ones we're going to talk about in our notes.
So I wanted to just kind of review a little bit, just pick up from where we left off last time. So last time we were together, we put this on the board. We said that the immune system has kind of two main parts. We have innate and adaptive, or specific and non-specific. The innate immune system we can split into the first and second lines of defense. So first line of defense is going to be like skin, mucous membranes, things like that, barriers to prevent things from getting inside our body. Most of this stuff isn't going to kill anything. It just protects us from the pathogens getting into us. Second line of defense is going to start killing stuff. This is going to have cellular parts like macrophages and neutrophils and basophils and eosinophils and natural killer cells and things like that. It also has non-cellular stuff. So complement system, inflammation, fever, things like that. And then the third line of defense is our adaptive or specific memory. And the third line of defense is made up of T cells, B cells, and antibodies. Now there's different types of T cells, different types of B cells. We'll talk about those. And there's different kinds of antibodies, but this is going to be what's going to make up the third line of defense. So as we look at this stuff, one of the points we made last time that's really important, first of all, is that all of this has to work together to provide immunity for our bodies. So if the first line of defense isn't healthy or working well, then there's going to be problems. You're going to get sick and you can die. If the third line of defense, if everything else is working perfect, but the third line of defense is having problems, or even just one part of the third line of defense, you can get sick and die. So we want everything to work together. Now, in order for that to happen, the immune system has to be able to recognize bad guys or foreign antigen and tell the difference between that and the good guys or self-antigen. So the foreign antigen are the surface features or the structures that are found on the surface of pathogens like bacteria or viruses or pollen or whatever that trigger our immune system. And our immune system has to be able to tell the difference between that and our healthy antigens like you would find on a red blood cell or a white blood cell or whatever. And it has to tell the difference between them. It wants to attack the bad guys and kill them, but not destroy our own body. All right. So first line of defense, mechanical barriers, unbroken skin, mucous membranes prevent the entrance of stuff into the body. Fluids wash away like tears, wash stuff away. And then second line of defense, we have inflammation. The tissue responds to injury that increases circulation, increases the movement of white blood cells to the site of tissue damage. We have natural killer cells, phagocytes, macrophages, fever. And so these are the kind of things we're talking about there with the first and second lines of defense. So third line of defense, we mentioned, third line of defense is going to have to tell the difference between self and non-self, and it does that by recognizing antigens. So non-self antigens would be like surface features on a bacteria like lipopolysaccharide or peptidoglycan or something like that, or a virus or whatever. Self antigen would be like the A antigen on a red blood cell, like an A red blood cell or whatever. So we have two types of adaptive immunity. We have cellular immunity and humoral immunity. And so this is going to be cell stuff and antibody stuff. And so like we said over there, it's going to be our T cells and B cells and then our antibodies. And so that's what we're going to talk about next. Okay. So when we talk about the third line of defense, all of the cells in the third line of defense come from the same place. They all are born in the bone marrow. That having been said, in order for these cells to actually function as immune system cells, they have to mature and they have to be trained, so to speak, to be able to fight off the infections. And so the T cells, after they're born in the bone marrow, they go to the thymus. We mentioned that last time. And then in the thymus, this is where they go through their selection process. So their maturation or their selection process happens there. Once they pass the test, after they mature, then they're released and go into the lymphocytes. They go into the circulation or they go to the lymph organ, to like a lymph node or the spleen or something like that. The B cells are also born in the bone marrow. They also have to go through a maturation process. Their maturation process happens also in the bone marrow. So they don't have to leave and go to the thymus, but they stay in the bone marrow, and that's where they go through their selection process. Once they pass their maturation test, then they go to the spleen or lymph node or something like that, or maybe into the circulation. But they're both born in the bone marrow, and then one of them matures in the thymus, and one of them matures in the bone marrow. Okay, so there are different types of T cells. And the three main types of T cells, the ones we're going to talk about in our class, are helper T cells, or CD4, cytotoxic T cells, or killer T cells, or CD8, and then memory T cells. So when we look at these three types of cells, they're all subcategories of T cells. And every single one of them is going to have an important aspect or important part of what the immune system is going to do. But in order for these T cells to work, normally what happens is, and it varies a little bit from situation to situation, but usually it's the helper T cells that get activated first. And so these T cells in our body, we don't want them all the time to be overly active, because if they're active all the time, then they're going to start attacking our own bodies. And so most of the time we want the T cells to be in a suppressed kind of non-active state. And then when something bad comes along, then we want the immune system to get excited really quick and then fight it off and then go back into its suppressed state so it doesn't attack our own bodies. So one of the ways that we do this is through the helper T cells. So there are second line of defense cells called macrophages, and sometimes other cells do this, but the second line of defense will eat stuff. And these macrophages, when they eat stuff, they'll tear apart the pathogen, and they'll take portions of the antigen on the pathogen, and they will attach it to a receptor, and they will show it to the immune system T cells. So what they're doing is they're presenting the antigen of the pathogen to the helper T cells. So these cells are called antigen presenting cells, or APCs. So antigen presenting cells, dendritic cells are one of the main kinds. Macrophages in general do it, and then sometimes B cells do it. And so when they encounter a foreign antigen and they eat it or they destroy it, they put portions of the foreign antigen on a special receptor called a major histocompatibility complex, or a human leukocyte antigen complex. And then they show it to the helper T cells. Now, when our bodies make T cells, helper T cells, they make thousands of different kinds. Our body will take the genes and they will randomly combine the genes together in different ways to make as many different types of T cells as they can. The problem is that sometimes it makes T cells against our own body. So we have to destroy those. That's what the maturation or the selection process is that happens in the thymus. But even then, it only makes one or two of every kind of T cell. And so what happens is the right T cell has to come in contact with the right antigen. And so the antigen presenting cell has to move through the lymphatic system or through a lymph node. It has to move through our body until it encounters the right helper T cell. And once they encounter each other, it's kind of like the key to your car. It will match the car and you can turn your car on. But if you have someone else's key, it doesn't work. And so the right key has to come together with the right lock. Once they come together, then you activate that helper T cell. And what's going to happen when the helper T cell gets activated is the first thing it's going to do is it's going to proliferate. It's going to divide. It's going to make more T cells of the same kind. So then we build up this large population of this T cell against this antigen, and so it's going to make us have a better army or a better set of defenses against it. The other thing that happens when the helper T cells get activated is the helper T cells release chemical signals called cytokines. And there are a couple different types, but the chemical signals that the helper T cells release when they're activated will do things like increase the ability of antigen presenting cells to present and to eat stuff, increase the activity of killer T cells or cytotoxic T cells. It'll activate these so they can kill stuff. It will increase, stimulate inflammation, increase white blood cell production. So the cytokines they release will go to the bone marrow and tell the bone marrow to make more white blood cells. They also will help activate B cells. We'll talk more about that when we get to B cells in a couple minutes. So this is really important. Now, the thing to remember about helper T cells is helper T cells don't kill anything. So these are kind of like dispatch, like emergency dispatch. So here, someone comes in and they try to rob me or, I don't know, steal my money. So I call 911. I say, hey, I need help. Well, the dispatch lady doesn't come from her office and run down here, or man, and run down here and beat up the guy who's trying to rob me. What does she do, or he? He calls the police. Yeah. So he calls the police, or he calls the fire department, or he calls the emergency vehicles, or whatever. That's what helper T cells do. they don't actually kill stuff, but they send signals that turn these on so they come and kill stuff and then make memory T cells as well. So that's an important thing to remember. So then the cytotoxic T cells also are dormant, more or less, until they get the signals from the T cells, or they get some kind of signal that makes them activated. Then when they get activated, then they're going to divide also. So we make more of those. In both of these types of cells, once they start dividing, they're going to make more of the same kind of cell that responds to the specific antigen that they're being activated to kill. They also will create memory cells. So what that means is then we create a large population of these cells in our body, and we retain those cells. So in the future, it doesn't take as long for me to go through the parking lot and find my car. They have the key, and the key has a whole bunch of cars that it can start. I don't know if that analogy works. Anyways, there's a whole bunch of those killer T cells and stuff, and so they can start and activate the system more quickly. Does that make sense? Okay. All right. So different kinds of cytokines. Cytokines are chemical signals that the immune system uses to interact with each other. So when you think about the immune system doesn't have a mouth, and it can't say, hey, come help me. It can't call on its phone. It can't wave its arms. They don't have arms. They don't have ears. They don't have mouths. So the way they communicate with themselves and with the rest of our body is through chemical signals that we call cytokines. So cytokines, there's different types. We have CSFs, or colony stimulating factors. We have interferons, or INFs, interleukins, or ILs, and tumor necrosis factors, or TNFs. And there's lots of different kinds of each one of these. In general, these are going to stimulate bone marrow to produce lymphocytes, like macrophages and dendritic cells. These are going to stop cells that have been infected by a virus from being able to multiply. Good. And they help kill cancer. These also help kill cancer. And there's, again, different types. And interleukins basically work between the white blood cells. So they're going to stimulate activation and proliferation and differentiation of the T cells and the B cells so that they can go and actually kill things. And again, there's lots of different kinds of each one of those, but those are general categories of each one of them. Okay. So that's kind of the T cell part. Now, again, there's a lot of detail to this that we're not going into, but that's at least kind of how they work and one way that they work. And that is really good to understand. It's also good to understand how the B cells help. Now, B cells do a couple different things. When B cells are activated, they also are going to divide really fast. They're also going to make more B cells of the same kind. They're also going to make memory B cells, just like the T cells did. The other thing that B cells do when they're activated is B cells make antibodies. Now, we'll talk about what those are and why those are important, but antibodies are a really, really important part of our immune system. And so we need to understand what those do, and we'll get to that in a second. They're also called immunoglobulins, same thing. Okay, so how do we activate B cells? Well, B cells can be activated in a couple different ways. We just mentioned one. One of the main ways that B cells get activated is when the helper T cells are activated and they release the cytokines. So cytokines from helper T cells will activate B cells. In some cases as well, B cells can activate themselves. So sometimes, depending on the type of foreign antigen that's infecting your body, sometimes B cells can actually bind to that foreign antigen, and that will stimulate them to start dividing and releasing cytokines also. So it depends. Either they activate themselves or they're activated by helper T cells when helper T cells are activated. Okay. So when B cells are activated, as we mentioned, they're going to divide really fast. They're going to make memory B cells, more B cells of the same kind, and they're going to make plasma cells. Now, plasma cells are the cells that make antibodies. And antibodies, also called immunoglobulins, have proteins called globulins that hook them together. They have 4 chains, 2 heavy chains, 2 light chains, and there's 5 kinds. There's immunoglobulin, or IgG, IgA, IgM, IgD, and IgE. E. And you can look at this list and see kind of what the main functions of those are. Now, when you look at these, one of the reasons why they kind of have these functions, part of it is because of where you find them. So the main types of antibodies that you find in the blood are going to be IgG and IgM. So IgG is really prevalent in the blood. You also find instead, IgA, you find IgA in exocrine gland secretions, so like the mammary glands and glands like that. IgE is also found in glandular secretions. IgE is the main antibody that stimulates inflammation, although others can do that as well. IgM, you also find that in the intestinal tract, so it's going to be involved in food and stuff. And so there's certain kind of patterns that you see to where each one of these are. But as we look at these, the general pattern of an antibody is like this. So you're going to see heavy chains listed in dark purple or purple. Purple are the heavy chains. Blue are the light chains. And the part down here is going to be consistent with the type of antibody we're talking about. So all IgGs are going to have basically the same heavy chain. And then they're going to have a variable region that's going to be made specifically for whatever the foreign antigen is that they're attacking. The blue part shows light chains, so constant light chains. And then there's going to be a variable part of the light chain as well. They're held together with disulfide bonds, so that kind of helps hold them together. But they're pretty simple. Some are bigger, some are smaller, some have different kind of patterns to how they look or whatever. But they're all going to have that basic same pattern where they have two heavy chains, two light chains, and part of each of those will be a variable region that will be made specific against a foreign antigen. Okay. So we already talked about that. All right, so why are antibodies so important? Well, antibodies are important because antibodies do a lot of things that help fight off infections. So this slide kind of talks about that. So antibodies, when they're released from a B cell, they can stimulate agglutination. So agglutination means that they're going to attach to a foreign antigen or to a pathogen, and then they're going to clump it together. So they clump it together just like putting shackles on a prisoner. If you put shackles on the prisoner, they can't run away. And so you clump them together, it makes this big clump of stuff, they can't escape, and then your body says, oh, there's this big clump of stuff, so I need to go kill it. So the macrophages come along and eat it. Precipitation, they can make antigens insoluble, and it makes phagocytosis easier. They stimulate neutralization, which means they cover the outside of the pathogen, and they prevent the pathogen from binding to our cells. So in a lot of cases, when you talk about a virus or you talk about a toxin from a bacteria or other things like that, one of the very first things that has to happen for those things to infect our body or cause damage to our tissues is they have to attach to our tissues. And if they can't, then they can't cause us problems. So when the antibodies bind to the outside of a toxin or the outside of a pathogen, it creates a barrier between those toxic substances and the target cell that they're trying to get attached to. And if they can't attach to it, then it protects us. So neutralization protects us. Antibodies also can activate the complement system. So the complement system is a system of proteins in our immune system, in our blood, that is not cellular, it's only proteins. And that system of proteins, when it's activated, will do four things. It will stimulate opsonization, which increases the ability for macrophages to eat stuff. Stimulate chemotaxis, which means that they will attract white blood cells to the site of the infection. They stimulate cytolysis, which means they will actually form a complex called a membrane attack complex, which stabs holes in the outside of the pathogen to kill it. And they can also cause neutralization. So they bind to the outside of the pathogen and prevent it from being able to attach to its target cells, just like we set up here. So antibodies can stimulate or activate the complement system, and that will cause all these things to happen. Antibodies can also stimulate inflammation, which I don't think is listed up here or down here. Yeah, right here. So they can stimulate inflammation. And so a lot of times when we think about inflammation, we think about problems associated with it, which there definitely are problems with inflammation. But inflammation is an important part of our immune system. And if it's kept in check, just like everything else, it can actually help our immune system and help our bodies. So if we have tissue damage of some sort, we increase the circulation to that area of tissue damage by releasing histamines, primarily from basophils and mast cells. And one of the ways that can happen is if antibodies bind to those cells, that triggers degranulation or the exocytosis of these inflammatory proteins. That increases circulation to that area, which brings extra oxygen and nutrients and white blood cells, and increases circulation to get rid of waste products. And so all that stuff works together to help the tissues heal and repair so that you can get rid of whatever the damage is. So antibodies do a lot of things. One of the other points, we kind of made this, but just to emphasize more, is that antibodies are very, very specific. And so not only do we have these structures, we have other parts of the immune system that fight stuff off, but the T cells and the B cells, the antibodies, when they're stimulated to fight off an infection, they're targeting one very, very specific thing. Now that's great for the immune system, but it's also really important because you can actually, as scientists have learned how these work and how they're built, you can artificially manufacture antibodies, and then you can attach like chemotherapy drugs to the one part of the antibody and make antibodies against a cancer cell or something like that. And so you can manipulate the system and use it for a treatment to help fight cancer or do different things, which is super cool. Okay, so when we talk about the immune system, basically what's going to happen when you have an initial exposure to an infection is you're going to get a primary response and a secondary response. And so the primary response is like what we said a minute ago, where the antigen presenting cell binds to the foreign antigen, it eats it, it rips it apart, and then it moves through your body, just like if I don't know where my car is parked in a parking lot and I have to wander around pushing the panic button until I find my car. Like it takes a while sometimes maybe, and they move around. Once they bind to the proper helper T cell, the T cell divides, releases cytokines, you stimulate the immune system. But this usually takes about 5 to 10 days for the immune system cells to interact with each other and to start producing antibodies. The secondary response, once you already have this stuff in place and you've created memory B cells and memory T cells and you have some antibodies in place, the second time you trigger that immune system, it happens super duper fast, 1 to 2 days, or maybe even less. And then you have antibodies that remain for months or years, and sometimes for the rest of your life. And it kind of depends on the different types of pathogen. And from person to person, it varies a little bit. But for the most part, if you get subsequent infections, your immune system will last for even longer. So these two graphs kind of demonstrate that. So initial exposure takes, I don't know, around 10 days or 15 days or so. It works really well, builds up, and it starts to drop off. A lot of times it will go back down to zero after some time. Subsequent exposures to the same foreign antigen, boom. The immune response is way faster than 15 days. It goes way higher and it lasts way longer. And so this is a really powerful tool, and it's something that's really neat. And so there's a lot of different sort of philosophies about, triggering the immune system or helping our bodies be healthy or whatever. And one of the ideas that people have come up with in the medical community is this idea of a vaccine. So in the case of a vaccine, what you're trying to do is you're trying to stimulate the initial response in a way that's not dangerous or lethal to your body. And then in some cases, you trigger the secondary response, or some cases, you just wait for your body to encounter the antigen and trigger the secondary response by itself. But if you can make that initial part at least in a way that's not dangerous to your body, then that can be really, really, really beneficial. And so overall, when we talk about the immune system, there's basically 4 ways that we can make the immune system help our bodies. One is called naturally acquiring immunity. One is called artificially acquiring immunity. One is called passive immunity, and one's called actively acquiring immunity. And so when we talk about natural versus passive, natural versus artificial, and passive versus active, these two things are going to go together. But what natural means is that you get exposed to the antigen in a natural way, like breathing germs or eating a spoon that had somebody sneezed on or whatever. I mean, not eating a spoon, eating from a spoon that somebody sneezed on or whatever. So kind of natural ways where the germs get in your body. Artificial means that it's introduced into your body in an artificial way. So this would be like an immunization or a gamma globulin shot. And then passive means that I acquire the antibodies without making them. So I'm giving them through a shot or through mom. And active means that I have to make them. So if I have naturally active, what that means is somebody sneezes on me, my body finds the antigen, the macrophages and the antigen presenting cells present, the T cells go through their process, they activate the B cells make antibodies, blah, 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 blah. So it's a natural way, I have to make them. Natural passive would be I get the immune system stuff, but I don't make it. So maybe a baby that breastfeeds from their mom. So breast milk has a lot of antibodies in it. Their IgG is small enough it can cross the placenta. So babies can get antibodies from mom through the blood circulation. So it's a natural way of getting it, but it's passive because the baby doesn't have to make it. And then artificial active would be giving me a vaccine. So I get the polio vaccine when I'm a little kid. My macrophages find the antigen, they eat it, they present, they trigger the T cells trigger the B cells, blah, blah, blah. Artificial passive would be acquiring antibodies through a shot, like a gamma globulin shot or immunoglobulin shot. And so then you acquire the proteins and it's already put together and it boosts your immune system, but you don't have to make it. And so those are kind of the four ways that we acquire that we get immunity in our bodies. Okay. So when we talk about the immune system, as we mentioned at the start, it's really important for the immune system to recognize foreign antigen and have to recognize it quickly and respond to it quickly. But it can't respond too much, or if it doesn't respond at all, then there's going to be a problem. So the two main problems we have with the immune system, if it doesn't work the way it's supposed to, is if it doesn't respond fast enough or doesn't respond at all, then of course we're going to get germs, we're going to get sick, and we can die. The other problem is if it responds too much. And so this slide here, in the next couple slides, are going to talk about hypersensitivities, hyper meaning too much, or high, like hyperactivity, too much activity. And so this can happen. There's 4 different categories of hypersensitivities or autoimmune responses. And this is when the immune system either responds when it's not supposed to or it responds too much. The first type is called type 1. The second type is called type 2. The third type is called type 3. And I'll let you guys guess what you think the fourth type is. No, it's type 5. I'm just kidding. It's really type 4. I'm just talking trash. Sorry. So type 1 is called immediate reaction. This is also called anaphylactic. Now, every single one of these 4 kinds of immune responses that are inappropriate have all kinds of different ranges of severity. So you can have a type 1 hypersensitivity that's really, really mild, and you can have a type 1 hypersensitivity that's life-threatening. And so normally when we think of anaphylaxis, we think of anaphylactic shock. But in reality, these can be really mild, something as mild as hay fever. So I don't know if any of you guys have hay fever or you have a minor allergy to, I don't know, something, dust or something like that. Yeah. So pollen in the spring makes you sneeze, makes your eyes itch. Like that's a type 1 hypersensitivity. And so it's not life-threatening. We might think it is when we're in the spring and our eyes are itching and we're going to crash into the car in front of us because you can't see or whatever. Or you sneeze a lot and people get mad because you're sneezing all over the place. But this kind of hypersensitivity is a problem, and the reason why it's a problem is because there's a malfunction in primarily IgE. And IgE will bind to the foreign antigen, of like pollen or a dust particle or something like that, and then it will bind to mast cells and basophils and stimulate inflammation, which increases mucus production and causes all those side effects. Now that's in a really small scale. In a real serious scale, then this would be IgE binding to lots of mast cells and basophils at the same time and causing them to release histamine all at once. And it causes vasodilation all over your body and inflammation all over your body at the same time. So it causes a decrease in peripheral resistance so much that your body goes into shock. And so then that would be like anaphylactic shock. And so there's all different levels in between as well. But that's a type 1 hypersensitivity. Type 2 is called antibody-dependent cytotoxic reaction. And this example of this would be if you have the wrong kind of blood transfused into your body. So then what happens is your antibodies recognize the wrong red blood cells as an enemy, because all it's looking at is the antigen, and they attack the red blood cells and rip them apart. And so that would be a blood transfusion. It also could be in a baby. If a baby is born in certain situations, sometimes the parents, because the blood types they have, can cause that to happen also, and it can kill the baby. In our day and age, it's not usually too big of a deal because your doctor will check that kind of stuff, and then they can give you a medicine that suppresses your immune system called Rogam, and so that's really common, and it usually works really well. But that's talking about here. So this is also a problem here. So the antibodies are attacking something we don't want them to, and so that's a problem. Type 3 is called immune complex. In this type of hypersensitivity, the antibodies bind to the foreign antigen like they're supposed to, but for some reason the macrophages don't kill it and rip it apart and clear it. And so then these clumps of stuff get stuck in your capillaries, or they get stuck in your kidneys, and that stimulates inflammation in those places. So it leads to things like lupus, it leads to things like glomerulonephritis, rheumatoid arthritis. It gets stuck in your joints, and then your body starts to fight off that stuff in your joints, and it kills your articular cartilage. And so those would be some examples of type 3 hypersensitivity. And then type 4, this kind of hypersensitivity is different from the other three in the sense that this doesn't involve directly involve antibodies or B cell products. This one involves T cells specifically. And so in this case here, what happens is it takes time for what's being presented to find the T cell that it's corresponding to and to trigger those T cells. And so that's the delayed reaction part. Examples of type 4 hypersensitivity would be like type 1 diabetes, multiple sclerosis. It also would be like cosmetic allergies or metal allergies, things like that. And so those are examples of hypersensitivities. Again, there's all different levels. Sometimes they just happen really fast and they're really serious, and sometimes they aren't very serious and they just go away. When you talk about delay cell, the reason why when you think about delay cell mediated ones, type 4 hypersensitivities, if you've ever had this experience, I don't know if any of you are allergic to like latex or like metal. When you first get the exposure, you may not even notice that there's a problem. But then maybe a few hours later, your hands start to itch or they get red or something. And then if you have like a ring that's like a metal that you're allergic to, then maybe a couple days later, you start to see blisters. And then a couple days later, you see it starts to bleed. And then it just gets worse and worse and worse over time. So that's kind of the idea, that it's delayed and it takes time for the immune system to respond to it more. All right, so that's a summary of this set of notes. We skipped a lot, but we also covered a lot. So if you guys have any questions, let me know.